anaerobic bacteria is a big family. It's not a family. It's a big group of bacteria. In this word, aerobic means that they prefer oxygen. While the first two letters, AN, anaerobic, means that they don't like oxygen. They prefer the condition without oxygen. Okay? So, in this big group, there are two categories, two big subgroups. One is the spore-forming anaerobes. Spore-forming means this group of bacteria, they can form spores. And we'll introduce the genus of Clostridium. Clostridium contains different species. We'll introduce three major species. So that's the Clostridium tetani, botulism, and perfringens. Those three different pathogens are so important. They can cause critical disease, or they can cause lethal the patient to die. And another subgroup of this big group is non-spore forming anaerobes. Non-spore forming, they don't form spores. And most of these subgroups are normal flora. So they are members of our normal microbiota. So usually they can live peacefully with us without causing any disease. Or sometimes they can cause some endogenous infection. So that's opportunistic infection. We'll focus on the first group of pathogens. We'll introduce some general knowledge in this genus before we take a break. First, we'll introduce some general properties. They are anaerobic, so that's for sure. And if you stain a bacteria with gram stain and check it with microscope, you can find this color, this blue or purple color. So they are gram positive. And the shape of the bacteria, they are bacilli. You can see bacilli. And they are relatively large. While compared to E. coli, they are much larger than E. coli. They are also motile, meaning that they do have flagella to help them to swim. They can form endospores, and that's for sure because they belong to the subgroup of spore-forming bacteria. They can produce toxins. So those toxins are so important. We'll introduce the toxins one by one. Their natural habitat is the soil or the intestinal tract of animals and humans. Natural habitat is the soil or the intestinal tract of animals and humans. Different species can cause different disease, including botulism, tetanus, gas gangrene, and also other diseases like colitis. So there are so many different species. And their morphology, and first they can form spores, but the location of the spores are different. Some are at the center, some are close to the terminal, what we call the subterminal. Some are exactly on the terminus, so they are terminal spores. Those spores are usually wider than the diameter of the rod, and that's one common sense. And you can see this bacteria. When the spores is formed by the end, the spores is always wider than the bacteria body itself. And they are motile because they can produce a lot of flagella. And they grow better under anaerobic conditions. For example, on blood-enriched medium. Many bacteria can produce a zone of beta hemolysis and blood agar. And we introduced beta hemolysis before. They can completely destroy the red blood cells. So the red color will be cleared out, and you can only see this yellowish color. So this is the general knowledge of Clostridium. And we'll have a break for about 10 minutes. And after the break, we'll come back to learn the first pathogen in this genus, the tetany, the Clostridium tetany. Okay. The first pathogen in this genus we are introducing is the tetany, the Clostridium tetany. And this is one pathogen which can produce a toxin called tetanospethamine. It's a very strong and toxic toxin. Tetany is spore forming, as we introduced. The spores is placed terminally by one end. So under microscope, you can see this drumstick-like structure. It's drumstick with one end swelling. So, and you can see different layers of the endospore. And they are obligate anaerobic bacterium. So that means that they don't like oxygen. They only prefer the condition without oxygen. So that is why Clostridium infection only happens in the deep tissue of our body. They don't cause infection on the epidermis, on the surface of our skin. They only cause infection in the deep tissues. They do not ferment sugars, and they do not digest proteins. So that is special because they must require some special nutrients inside of our body rather than using sugars and proteins. Here is how the tetany specimen, tetany toxin cause disease in human. This is a normal model of our human nerve cell and muscle cell interaction. The motor nerve cell can release this green molecule, and that's the acetylcholine. And this is a regulator or a neuron transmitter. And once this molecule binds to the receptor on the muscle cell, the muscle will contract. So this is a contracted muscle. But you don't want your muscle to be contracted all the time, and you need to regulate the behavior of the muscle. Sometimes you need it to contract, and another moment you need to be relaxed, so that can control your action. So here is another neuron we call the inhibitory neuron. The inhibitory neuron can release a transmitter, the red one, and this red transmitter can tell the motor neuron to stop releasing the acetylcholine, and that will reduce the amount of green transmitter, so the muscle will relax. So this is the regulation mechanism of the motor inhibitor and neurons, and also the muscles. So this is a normal condition. But once there is toxin from tetany, so this is another slide. This is the toxin from tetanus. This tetany toxin, exotoxin, can bind to the inhibitory neuron and block the release of the red transmitter. So that means at this moment, the motor neuron cannot be inhibited. The motor neuron will release the green transmitter all the time, the acetylcholine all the time, and that will bind to the muscle cell and let the muscle cell to contract all the time. So the muscles cannot relax. And that is the mechanism how the tetany exotoxin cause disease. Okay, so this shown in this cartoon, in these slides. I have a video to show you the mechanism of tetanus specimen. And this is one of my favorite video showing the mechanism of a toxin. I'd like to show you the video first. It explains how the toxin works, how the toxin causes disease.
Tetanus is an acute, often fatal disease caused by a toxin produced by the bacterium Clostridium tetani. The toxin attacks the central nervous system, causing convulsive spasms of the muscles. Symptoms include lockjaw, stiffness of the lower limbs, and contraction of the neck and spine into an arch. These spasms are extremely painful and can cause bone fractures, tendon ruptures, and dislocations. Death often occurs due to respiratory failure or cardiac arrest. In newborns, the infection can enter via the umbilical stump in unhygienic conditions. Tetanus can also develop as a result of a deep puncture wound, as caused by stepping on a nail. It cannot be spread from person to person. Here we see the nail has entered deep into the muscle layer of the foot. The nail has introduced tetanus spores into the wound. Under conditions of low oxygen, as found in a deep puncture wound, the tetanus spores can germinate to produce living tetanus bacteria. The spores also release a toxin called tetanospasmin, which enters the bloodstream. Bacterial numbers increase as they grow and divide. Some bacterial cells develop spores at one end. The bacteria lies, releasing these spores and more tetanospasmin toxin. Bacteria without spores can also lyse to release toxin. The toxin enters the bloodstream. Movement is enabled by skeletal muscles, and skeletal muscles are controlled by motor neurons. An electrical message is sent down the axon of a motor neuron and arrives at the terminal buds. Chemical neurotransmitters are released, which bind to the muscle fibers, causing them to contract. Shortly after, the neurotransmitters are released from the muscle fibers, allowing them to relax. Repetition of this process tells the muscle fibers to contract and relax as required. If you are infected with tetanus bacteria, the tetanospasmin toxin can reach motor neurons via the bloodstream. The toxin enters the motor neuron via the terminal buds. The toxin then travels up the axon towards the neuron cell body, which is found in the spinal cord. After 2 to 14 days, the toxin reaches the motor neuron cell body in the spinal cord. Other toxins travelling in the bloodstream also enter motor neurons and make their way to the spinal cord. We will now zoom into the spinal cord tissue. In the spinal cord, the motor neurons are controlled by inhibitory interneurons. These interneurons prevent the motor neurons from constantly firing. Tetanospasmin toxins arriving in the motor neuron cell body interfere with the action of the inhibitory interneurons. To see how the toxin exerts its effect, we will zoom in on a synapse between a motor neuron and an inhibitory interneuron. The interneurons release inhibitory neurotransmitters into the synaptic junction. These prevent the firing of motor neurons so that the muscle fibers they control do not constantly contract. Tetanospasmin toxin moves out of the motor neuron across the synapse into the inhibitory interneuron. The toxin prevents the release of inhibitory neurotransmitters from the interneuron. The interneuron can no longer control the motor neuron, so the motor neuron fires constantly. Here we see many motor neurons have lost their inhibitory control and are constantly firing. This leads to continual release of neurotransmitters from the motor neuron terminal buds, which cause the muscle fibers to contract repeatedly until spasm occurs. The end result is the severe muscle spasms typical of tetanus. Tetanus is rare in most developed nations with comprehensive vaccination programs. With adequate immunisation, tetanus is a completely preventable disease. However, in the developing world, where vaccination is uncommon, tetanus is one of the top 10 causes of death. Many organisations are working to increase access to vaccination in developing nations, including vaccination during pregnancy and access to hygienic birthing conditions. You can protect yourself against tetanus by maintaining adequate immunity into adulthood by having a booster shot at least every 10 years.
Okay, so that's the mechanism of tetanospasmin. And later, this one, this text also tells us the same story of tetanospasmin, how it causes disease, how it works. Because we introduced the material in the slides, in the cartoon, and also in the video, we won't go over it again. And those detailed information statements should be available in your textbook. So we'll just skip this one. For pathogenesis of tetany, it's not an invasive organism. As you can see, they don't have flagella. They cannot invade actively in the human, but they can cause infection by accident. If there is a deep puncture of the body, they can get into the deep tissue where the condition is suitable for the germination of the spores. And the disease is almost entirely a toxemia. This word toxemia. Emia means something in the blood. Toxemia means toxin in the blood. So disease is mainly due to the production of toxin. And it's a neuron toxin and can cause a problem in our nervous system. And also the germination of the spore and development of vegetative organisms that produce toxin are infected or aided by other factors, including the necrotic tissue, like the calcium, salts, and also associated with pathogenic infections. All of these factors aid the establishment of low oxidation reduction potential. So they produce the condition of low oxygen condition, which can help the germination and growth of the bacteria. Okay, So that is the pathogenesis of tetany. And here are the clinical findings. The incubation period might be 4 to 5 days to many weeks. So it varies. And if you have a patient coming in showing the symptoms, you should ask the patient to tell you their injury history and for a long time, and to figure out whether it's due to some tetany infection. And tonic contraction is the major symptoms, and it can cause infection in either newborns or also in adults. Okay, so that's the clinical findings. And for diagnosis, and this time, because it's a deep tissue infection, we usually don't collect the specimen because it's not easy. And we usually diagnose by the symptoms if it's shown like typical tetanus symptoms, or we ask your patient about their history of injury, like some days ago, a couple of weeks ago, whether they got a deep puncture like this one, like a puncture of a nail through the foot. And if it happened before, it's possible that it can bring in tetany spore, and the spore can germinate inside the tissue. Prevention and treatment, we can use active immunization with toxoids. And we introduced toxoids before. They are originally toxins, but we can treat the toxins with formaldehyde so that we can remove the toxicity, but keep the antigenic property so that it can still stimulate the immune system to produce antibodies. And we should take care of the wounds properly. And if especially when the wounds are contaminated with soil, for example, we need to clean the wounds thoroughly. Sometimes you need to give your patient some surgery to open the wound and remove any debris in it. So this we call surgical debridement. And if it's urgent, we can also give the patient some antitoxin. So that's the antibodies or antiserum containing antibodies. Those antibodies injected to the patients can neutralize the toxin before they fix to the nervous tissue. So if there is some free toxin, the antibodies can bind and neutralize the toxin so that the toxin cannot bind to the nervous tissue, the nervous synapse, and cause disease. And you can also give your patient some penicillin to kill the vegetative growing bacteria. That will also help. To control tetany, we should rely on vaccines. We can use active immunization with tetanus toxoid. So that's the immunization. When the baby is born, we usually give them DPT vaccine. That contains 3 different components. D is the diphtheria toxoid, P is pertussis toxoid, and T is for tetanus. And this DPT vaccine is given to all children during the first year of life 3 times, 3 injections. And then there is a 1-year gap, and 1 year later, you give the children another injection. And this will set up immunization for the kid and protect them for several years, or about 10 years. So that's the plan. That's the plan in China and also in America, I think. I don't know the plan in your country. Maybe you also get those injections when you are young. So that's the active immunization. But in some countries, such as in Japan, after the initial immunization, when the baby grow up, when the children grow up, they give them booster injection upon entry into school. And even after that, even for adults, they will get a booster every 10 years. So that will keep them protected lifelong. That's not given in China, as I know, and not given in America. Maybe it's carried out in your country. Maybe you can do some research and get the information. For some people who are working in for special professionalism, and they might have a higher risk of exposure to tetany. And before they get the job, before they work, they should get the vaccine again so that they will get protected. So in the future, when you are a doctor, you should help your patient or help your guests to help them get protected. So this is the control of tetany. Okay. So next pathogen we are introducing here is Clostridium perfringens. That's a different pathogen in this genus. They are also forming spores, but the spores is placed subterminally. It's not exactly on the terminus, but close to the terminus. They produce capsule. As you can see here, you can see the white halo around the bacterium. So that's the capsule. The capsule can protect the bacterium, is one of the important virulence factor. They are not motile, means that they don't produce flagella, and they can produce a double zone of beta hemolysis. We learned beta hemolysis before. That means they can lyse red blood cells completely on the blood agar. But for this one, it's a special beta hemolysis. In the middle, there is one clear zone. This zone, we call it inner cytosome or hemolytic cytosome. And there is also an outer zone. The outer zone is not like complete lysis. That's because of another toxin, alpha toxin. They look different. So keep in mind this double zone of hemolysis, beta hemolysis on blood agar. It's due to the perfringens. It's a very specific characteristic. You can use this information for diagnosis. They can ferment a varieties of sugars and produce acid and gas. So they can utilize sugars. They can also digest proteins. So that is why this pathogen is so powerful, because they can use a lot of nutrients in the host. And if we culture the bacteria in a milk medium, so that's a medium containing milk. And in milk, there is a large amount of proteins and also sugars. And they grow well. And when they grow, they produce a large amount of acid and gas. And the gas will form a lot of bubbles. So bubbles or foams and can push the medium up. And the acid will denature the proteins. So the proteins will clot. So that's a very special characteristic. And we call this reaction stormy fermentation because it's so vigorous. We call it stormy fermentation. This is one typical experiment to diagnose perfringens. Okay. There are toxins and enzymes. We introduced the alpha toxin. So that's the one to form the outer hemolytic zone, outer beta hemolytic zone. Because this is alpha toxin is one enzyme called lecithinase, and the lecithinase can hydrolyze lecithin. So that's a component of the cell membrane and can cause lysis like this. While the inner membrane is the cytotoxin. That's the has hemolytic and necrotizing effect, but it's not the same enzyme of this lecithinase. It can cause complete lysis of red blood cells. Except for this hemolysin, they can also produce DNase. That's an enzyme which can degrade DNA molecules. You know, we introduced before that DNA molecule actually is very sticky, and so if they can degrade DNA, they can cut DNA molecules into small pieces. That can help the bacteria to escape from one place to another, help them to spread. They can also produce collagenase. Collagen is a type of important protein in connective tissue. And if they can produce collagenase, it can degrade collagen so that it can make gaps between the tissues. So they help the bacteria to spread within tissues and muscles. So that is why this pathogen is so powerful, so toxic. And they can produce enterotoxin. And this enterotoxin will function if we ingest large number of perfringen. They can cause infection in our intestinal tract and can cause intense diarrhea. But usually, even the diarrhea is intense, it's not life-threatening, and the patient will recover soon. And here is the detailed information of the pathogenesis of the bacteria. We won't cover it in detail, and you can follow these lines and read the material in your textbook to help you master this pathogenesis. Okay. Clinical findings. There are two types of infection. One is called the gas gangrene, like this. This is one the tissue recovered from gas gangrene. You can see the large area of damage in the human tissue, and it's deep in the tissue. They can contaminate the wound and cause infection in the deep side of the wound. And the progressing is rapid and causing necrosis, fever, hemolysis, toxemia, shock, and death. And you need clear the wound thoroughly and help your patients recover. So this is one of the most important disease it can cause. Another one is food poisoning. We introduced that they can produce enterotoxin, and once you ingest it, you ingest the bacteria, it can cause infection in the digestive tract and can cause diarrhea, but without vomiting and fever, and that will last for about a couple of days. But it's not life-threatening if it's ingested. For diagnosis, we can follow the standard procedure, collect the specimens, either the material from the wound, the pus, or the tissue, or you can collect the patient's stool and check it. And under microscope, they are gram-positive rods and very large. And if you isolate and culture the pure bacteria on agar, you can use different agar, including blood agar, chopped meat glucose medium, milk medium. So that's the stormy fermentation we introduced before. Or you can culture them on the egg yolk agar. So this is one medium containing egg yolk. Because in the egg yolk, it contains a lot of lecithin. And the bacteria can produce lecithinase. It can lyse, degrade the lecithin. So you will see a halo around the colony, around the area of the bacteria colonies. They make it opacity. So this is a special phenotype which can help you to diagnose the bacteria. Okay, so this is the laboratory test. And treatment, we introduced that because they cause infection in the deep tissue, we should use a surgical debridement to remove the bacteria from the deep tissue. And we can give the patient with antimicrobial drugs, particularly penicillin, to kill the live bacteria. And you can give your patient high level of oxygen because this bacteria is anaerobic bacterium, and high level of oxygen can inhibit the growth of bacteria. We can also use antitoxin to neutralize toxins produced. For the food poisoning caused by perfringen, they produce enterotoxin and can cause diarrhea. We usually give the patients some symptomatic care. So that's a supporting treatment. And the patient usually will recover with, it's not life-threatening. So this is treatment. Okay, so I think we'll just stop here and we'll leave a third pathogen, the botulism, for the next time. And next time we'll finish botulism and we'll introduce Mycobacterium and also another category of pathogen. So this Thursday we'll finish bacteriology part. And from next week we'll learn virology. Okay, that's all for today.